Welcome to IGCSE chemistry lessons. In this video, we are looking at common apparatus found in the chemistry lab. In our introduction, let us define what is meant by the term laboratory apparatus. Laboratory apparatus refers to the various tools, equipment, and instruments used in scientific laboratories to carry out experiments and research. These apparatus play a vital role in the accuracy and reliability of scientific data, ensuring that experiments can be replicated and verified by other scientists. In this regard, the purpose of this lesson is to explore the sources of heat in the lab and the apparatus used to determine volume of liquids and gases in the lab. The first source of heat used in the lab is a Bunsen burner. A Bunsen burner uses gas to light up a flame. This one is a source of heat, so it is connected. It's got a gas source here which is connected to a gas tank, and then it has got a gas valve here, the one which is used to regulate how much gas should be coming through, and then we have the air hole here, which also can be used to regulate how big the flame is being produced. So this gas burner or Bunsen burner is used as a heat source in the absence of flammable materials. Please take note, this one should be used in the absence of flammable materials. Let's say an experiment is using ethanol. So if it is using ethanol, then that particular experiment shouldn't be exposed to a Bunsen burner because it has got some naked flames. A Bunsen burner produces a single open gas flame and is used for heating, sterilizing, and the like. How a Bunsen burner is used? It is used alongside what we call a tripod stand. A tripod stand has got a wire gas on which you can put anything that you are heating. Commonly, we heat things in a beaker. So you are going to put things in a beaker. For example, you want to heat some water, you want to boil some water in the lab. So you're going to put water into the beaker, and then you set up your Bunsen burner like this on the tripod stand, and then the wire gauge is put on top. So we are saying a laboratory tripod stand is a three-legged platform used to support flasks and beakers during heating. Another source of heat in the lab is what is known as a hot plate. A hot plate is a device that consists of a flat plate attached to an electronic heating mechanism. The main aim of a hot plate is to evenly heat a substance or sample placed on top of it. Now, this is how a hot plate will look like. It has got the heating knob. Okay, it has got the heating knob, it has got the speed knob, then it has got the hot plate itself, and on top of the hot plate, that is when you can physically put whatever it is that you want to heat. Now, judging from this one, you need to make a choice based on the experiment that you are doing. What source of heat should I use? So that will depend on what materials you are using. So if you are using anything flammable, then you would rather use a heat source. If you are using something that is, if you are using anything flammable as your materials in your experiment, then you would better use a hot plate. But non-flammable things, you can use a Bunsen burner. But also one advantage that this one has is that it's able to evenly heat. But a Bunsen burner will not evenly heat because there will be the beginning of the heat. Let's look at the hot plate versus a Bunsen burner. So we are saying the advantage of using a hot plate in place of a Bunsen burner is that a hot plate does not make use of flammable fuel, thereby minimizing the chances of what? accidents. So a hot plate does not use flammable fuels. Therefore, you minimize chances of accidents as opposed to a Bunsen burner. The temperature of the Bunsen burner cannot be determined easily, but the temperature of the hot plate gets recorded and displayed in digital format on the indicator attached on the device and can be controlled easily. Remember, we have a knob. You can determine how much heat you want to be coming out, and also the speed at which it can be done. So that is an advantage. So two of the advantages is that a hot plate does not use flammable fuels, and then we know that the temperature of the hot plate can be controlled. But apart from that, also the third one you can add, which is not written down here, is the fact that a hot plate is able to evenly heat, okay, evenly heat the material. So there are about 3 advantages that has been mentioned. Take note of them. Let us now look at the apparatus used for measuring volumes. So first of all, you need to remember that volume is defined as the space occupied within the boundaries of an object in 3D dimensional space. It is also known as the capacity of an object. Okay, so how much can you put in something? If you have a container, how much can that container hold? So the how much is actually the volume that we are talking about. So the first thing is we have our measuring cylinder. We also have our burette. Then we also have our pipette. And then we're going to have the graduated syringe. Okay, so this one is a graduated syringe. So these are the apparatus for measuring volume. Let us look at how exactly they are used. But before we do that, let's also look at some supporting apparatus as you are measuring volumes. So we have a funnel, and we are saying a funnel is a conical utensil having a small hole or narrow tube at the apex and used to channel the flow of a substance into a small mouthed container. Now, when we say container, we don't just mean the containers of cooking oil, the containers of what? A container simply means something that can contain a substance. So it could be even in the lab, we can talk about containers, we can talk about beakers, we can talk about test tubes, we can talk about burettes, we can talk about pipettes. All those are containers with respect to the lab. So container is simply something that contains, that is able to contain a substance. Now, some containers, they are small mouthed. They are small mouthed. So how do we put in a liquid inside that container? Is by using the funnel. So this funnel, you can just put it, insert it. Let's say this is the mouth of the container. So this is the container that you have. So you can just insert it on top of the container, and then you start pouring the liquid through here, and the liquid will be able to go through. Very important. So in simpler terms, a funnel is used to guide liquids and other substances into containers, for example, during the filtration process. Let's look at how to read volume. Very important. As you do your practicals, as you do your alternative practical final examinations, this aspect is going to be very important. So we are saying when you are dealing with, when you are reading volumes from tubular apparatus such as burettes, pipettes, and measuring cylinders, etc. What you are supposed to do is, I'm going to explain this in a short while. When you have a small or a tubular apparatus, when you pour in a liquid into this tubular apparatus, what is going to happen is the water is going to form a curve, or the liquid in general, it forms what we call a curve. So this curve here, so it will be a curve that is formed like this. It curves inwards, and this curve is what we call a meniscus. So the curve is called the meniscus. So the curve is called a meniscus. Now, this meniscus has got 2 parts. It has got what we call the upper meniscus. So we have the upper meniscus, and then we have here where it bends at the bottom. That one we call it the lower meniscus. Okay, so now how do you make the readings? The readings are supposed to be taken from the lower meniscus. So you don't look at the upper meniscus, but you look at the lower meniscus. That is where the volume readings are taken. Let's look at an example here. So this is the liquid that was poured into this measuring cylinder right here. And remember, our apparatus that are used to measure volume, they are graduated, meaning there are scales that are marked on them. So the first thing that you're supposed to do is you're supposed to figure out how the scale is being calibrated. So for example, I can see this long one here. The long one here is 19. Then I'm going to check, okay, if this one is 19 and then this other long one here is 20. So how are these divisions being counted? So can I try one? So if I say 19, 20, it's not going to make sense because the 20 is already here. So if one is not working, then I'm going to try 0.5. How about if I say this is 19.5, okay? Then again, be 20. So 0.5 is not working. Then let me try 0.1. So if this one will be 0.1, this one will be 0.2, this one will be 0.3, this one will be 0.4, this one will be 0.5, this one will be 0.6, this one will be 0.7, 0.8, then 0.9, and then 20. So the 0.1 has made sense. So you can check it out, quickly check it out and see if the divisions. Now each division can either be 1, can be 0.1, can be 0.2, can be anything. So never take it for granted that you're just supposed to memorize. Always check for these things. So here you have checked that the calibration is 0.1. So now look at the lower meniscus. If you check at the bottom here, the bottom is actually, the lower meniscus is on 20. So meaning the reading here will be 20 mils or cubic centimeters. Now the upper meniscus is slightly somewhere here on top, which will be somewhere at 19.8. And this reading here is going to be wrong. So if you take the reading from the upper meniscus, it's going to be wrong. The correct reading is supposed to be from the lower meniscus. That is what is supposed to be done. Please take note of this every time you're dealing with any apparatus that is tubular and is able to form the lower meniscus. When you put a liquid, always take the readings from the lower meniscus. All right, let's now look at some of these apparatus. So the first one that we have is a measuring cylinder. Of course, it has got different sizes. Now, we are saying a measuring cylinder is a common apparatus found in the lab. It has markings on its walls from where the volumes are read, and this is why we say it is graduated. So when we say something is graduated, meaning it is marked with a scale. So it is used for making approximate. Please take note, it is used for making approximate measurements of volumes of liquids. Approximate measurements. Volumes of liquids are taken from the lower meniscus, as earlier discussed. Then measuring cylinders are of different sizes. Then we also have a burette. Now a burette, it has got a tap here and it has got markings. So the markings here will begin from 0 and they'll end all the way up to the 50. That is the most common burette type that you're going to have. Now, the way the burette is used is it has to be mounted on a clamp stand. So this one you're seeing here is a clamp stand. So it has to be mounted on a clamp stand, and then the liquid to be measured, it can be put in here, okay? And this is usually a titration setup, but for now, what you have to know is a burette, if you want to use it to measure volume, it has to be mounted on a clamp stand as shown in this diagram. Now, a burette is used for accurate, so please be taking note of the differences. For the measuring cylinder, we said it is used for measuring approximate, for making approximate measurements. But for this one, we are saying accurate measurements of different volumes of what? Liquids. It can measure small amounts of volume. For example, a burette can be used to measure exactly 22 cubic centimeters of a liquid. So the bottom line here is this one is used to measure accurate measurements of different volumes of liquids. Okay, so that is a burette. It has a scale which starts from 0 cubic centimeters at the top up to 50 cubic centimeters at the bottom. So the scale is more sensitive than the measuring cylinder. So when you are carrying out an experiment, you need to choose an appropriate apparatus. So if you are looking for accuracy, you would rather get a burette and not a measuring cylinder. Let's look at a pipette. Okay, so this is a pipette. We also have different types of pipettes and different sizes of pipettes. Okay, so now we are saying a pipette is used for delivering accurately a fixed volume of a liquid. So we started from measuring cylinder. A measuring cylinder does approximate measurements of volumes of liquids. Then we went to the burette. A burette measures the accurate measurements. Then we go to a pipette, the one which we are saying, it is used to deliver accurately a fixed volume of a liquid. Now, those are the differences. Now, what do we mean accurately a fixed volume of a liquid? If you are using a pipette which is labeled 1 ml, meaning it can only measure 1 ml. If you are using a pipette that is labeled 5 ml, it can only measure 5 ml. Nothing less, nothing more. That's the way it works. If you are using the one which is labeled 25 ml, meaning it can only measure 25 ml. So when you are here on the mark here, that is where the 25 is. Here on the mark here, if you're using a 5, that is where the 5 is. And then there is also here, it is nicely labeled. So the 1 ml is actually labeled nicely here. Now the question is, how do you put a liquid into the pipette? How to put a liquid into a pipette? So we use what is known as a pipette filler. Okay, we use what is known as a pipette filler. Now, the common pipette that you're going to see is as displayed here. So this pipette has got parts. It has got A here, where you are seeing here, there is an A, and this A is just for air. So A is for air, and then it has got an S here. This S is just for S valve is used for sucking the liquid, okay, for sucking the liquid. Then we have got an E. So this E is just for emptying, okay, for emptying. So now the way here, this side, this other side here, that is where you insert the pipette. This is where you insert the pipette. So what are you going to do? You are going to insert the pipette there. You insert it. Then when you insert the pipette, you are going to squeeze the air, okay? You're going to squeeze the air, and then you are also going to squeeze this region here, which I like to call the tummy. So you're going to squeeze the tummy here, and you squeeze it at the same time. Air and the tummy, you squeeze it at the same time, and then this will become flat. When it becomes flat, and then you are going to insert into the liquid. So when you insert into the liquid, then you are going to squeeze. You are going to squeeze on the S valve. When you squeeze on the S valve, then the liquid will start getting into the pipette. That is how it is done. Okay, there is also another simpler version of a pipette which just has a wheel. So the procedure is the same. Here is a pipette that is inserted. You put it into the liquid you want to draw, and then you just start moving the wheel. So you start moving the wheel. As you are moving the wheel, the liquid is coming up. So this one is a little bit simpler. Now we look at the gas syringe. So this is called a graduated gas syringe. Now the volume of a gas can be collected and measured using a graduated measuring apparatus called a gas syringe. So this gas syringe can be used to measure the volume of a gas. So a graduated cylinder inverted into water may also be used provided the gas isn't soluble. So we have an alternative here. So if you don't have the gas syringe, then you can also use a graduated cylinder. You invert it into water, and it will be able to collect the gas, but this will be dependent on the nature of the gas. If the gas isn't soluble in water, then well and good. So look out on the collection of gases. Okay, look out for the video on the collection of gases. So if the gas happens to be heavier than air and is colored, the cylinder can be used upright. Let's look at how this thing is done. So you are going to put a conical flask. You are going to make sure it is airtight here as you put the straw, okay, and connect the syringe. So the syringe has got the plunger here, and then whatever reaction is producing the gas here, the gas will have no choice to escape in the site because it's airtight, it has been sealed. So the only place where the gas will go is it will go into this straw and finally get into the what? Into the syringe. Now the syringe is going to be pushed by the pressure. So as it moves, there are readings that are on the side here. So as if the gas moves as much as here, it means here where it has reached, that is how much gas has been produced from here. If it reaches there, then it means that is how much gas has been produced from here, because the gas has got nowhere else to go other than just coming through. Thank you for this lesson. Look out for part 2 on other general apparatus.